Good morning, everyone, and welcome. I am delighted to have you here today. Today, we will be exploring the topic of working standard management, led by Dr. S.K. Singh, who holds a PhD in this field. In this presentation, we'll cover the key concepts, best practices, and strategies to effectively manage and maintain working standards in various environments. So, let's get started and take a look at what's ahead. Working standards, or WS, are secondary reference materials that are prepared from qualified sources. These standards are essential for routine analytical testing and assay determination, ensuring consistency and accuracy in our results. It's important to note that WS must be traceable to official reference standards, like those from USB, EP, BP, or IP. This traceability guarantees the reliability and validity of our testing processes. Additionally, we need to follow good manufacturing practices, GMP, along with ALCOA Plus principles and data integrity guidelines. All procedures used to prepare and handle these standards must be validated to maintain their quality and trustworthiness. In summary, proper management of working standards is vital for maintaining the integrity of our analytical work and ensuring compliance with regulatory requirements. Ensuring consistent and reliable results in analytical testing is absolutely essential for maintaining quality and confidence in our data. To achieve this, we need to maintain traceability to primary or qualified reference standards, which serve as the foundation for accuracy. Establishing standardized procedures for preparation, qualification, and storage helps us minimize variability and ensures everyone follows the same rigorous protocols. Controlling the potency, purity, and stability of our working standards is also critical, as it directly impacts test accuracy. Regular monitoring and requalification of standards help us minimize variability over time and keep our results dependable. Compliance with regulatory expectations such as ICH, WHO, and GMP is non-negotiable. These guidelines ensure our methods meet global standards. Throughout the life cycle of our data, maintaining data integrity and thorough documentation is key to transparency and traceability. Supporting laboratory efficiency involves making sure that qualified working standards are readily available when needed. Overall, the objectives of working standard management are centered on ensuring reliability, consistency, and compliance across all aspects of our testing processes. Let's take a closer look at some of the key regulatory standards and guidelines that shape our pharmaceutical quality systems. Starting with ICH Q6A and Q6B, these provide specifications and analytical procedures essential for ensuring product quality and consistency. They help us define clear methods for testing and characterization. Moving on, ICH Q7 focuses on good manufacturing practices specifically for active pharmaceutical ingredients, or APIs, emphasizing the importance of controlling manufacturing environments and processes. Then we have ICH Q8, Q9, and Q10, which cover pharmaceutical development, risk management, and the pharmaceutical quality system itself. These guidelines help integrate quality into every stage of development and manufacturing, fostering a proactive approach to risk and process control. The WHO Technical Report Series offers international standards on reference standards and quality control, which are vital for maintaining global consistency. On the regulatory front, the U.S. FDA's CFR Title 21 Part 211 outlines laboratory controls and documentation requirements, basically, the backbone of compliance in the U.S. USB, EP, and IP provide recognized standards on reference and working standards, ensuring uniformity across different regions. ISO IEC 17025 sets the requirements for testing laboratories, emphasizing technical competence and quality management. GXP guidelines, encompassing good practice principles, guide us in managing, qualifying, and handling our processes and equipment properly. Altogether, these regulatory references form a comprehensive framework that underpins our quality assurance efforts, ensuring that medicines are safe, effective, and of consistent high quality across the board. Let's start by talking about primary or reference standards. These are typically obtained from well-established sources like pharmacopoeias such as USB, EP, BP, or IP. They serve as the benchmark for accuracy and quality in our testing. Moving on to secondary or working standards, these are derived from the primary standards and are used routinely in the lab. They are qualified against the primary standards to ensure their reliability and consistency. Then we have impurity standards, which are used to identify and quantify impurities in the drug substances or products. These standards help us maintain the purity and safety of our medicines. Additionally, some labs develop in-house standards or custom standards. These are created when commercial standards are unavailable or specific testing requirements demand tailored solutions. Now, let's connect these concepts to the overall theme of quality control in pharmaceutical analysis. Understanding the different types of standards, primary, secondary, impurity, and in-house, is crucial for ensuring that our testing is accurate, reliable, and compliant with regulatory requirements. Each standard plays a vital role in maintaining the integrity of our quality assurance process. Let's talk about the key qualities that make for effective working standards. First, high purity is essential, typically at 99% or more, which ensures that the standard is reliable and consistent. It's also crucial to have proven identity and potency, so we know exactly what we're working with and that it performs as expected. Additionally, the standards must be free from significant impurities and moisture, because contaminants can affect the accuracy and reproducibility of our results. Stability under defined conditions is another vital point. Our standards need to remain consistent over time without degrading. Lastly, a validated qualification procedure is necessary to confirm that the standards meet all these requirements reliably. Together, these qualities form the foundation for trustworthy and effective working standards, which are central to maintaining quality and confidence in our analytical processes. Now, let's look at how we source and prepare our working standards. First, we have in-house API batches, which means we produce some of our active pharmaceutical ingredients internally, giving us greater control over quality and consistency. Next, we rely on supplier-provided primary materials, ensuring we have high-quality starting points from trusted sources. It's also crucial that we qualify these materials according to pharmacopoeial standards, which helps us meet strict regulatory requirements and maintain confidence in our processes. When necessary, we isolate and purify specific compounds to serve as precise standards for our testing. All of these sources, from in-house batches to purified compounds, ultimately form the foundation of our working standards, which are essential for accurate and reliable quality control throughout our development and manufacturing processes. This integrated approach helps us ensure consistency, compliance, and ultimately the safety and efficacy of our products. Now, let's walk through the key steps in our qualification process. First, we have sample selection and approval, which is crucial because choosing the right samples ensures the integrity of the entire process. Next, we perform testing against a reference standard to confirm that our samples meet the expected quality criteria. Following that, we conduct an analytical data review to thoroughly evaluate the results and verify consistency. Once we're satisfied with the data, we move on to potency assignment, where we determine the correct strength or concentration of the product. Then, we plan the stability studies, which are essential for understanding how the product maintains its quality over time under various conditions. After all this, we seek approval and labeling, making sure everything is compliant and accurately communicated to users. Finally, we implement controlled distribution to maintain product integrity throughout the supply chain. This entire process forms a comprehensive qualification overview, ensuring that every step supports the safety, quality, and efficacy of our products from start to finish. Let's dive into the identification methods used to confirm the compound's identity. We typically rely on IR, UV, and HPLC techniques. Each of these plays a crucial role in ensuring we're accurately identifying the substance. Moving on, The assay by a validated method is essential to determine the correct potency and concentration. This helps us verify that the product meets its specifications. Next, we have HPLC purity, which assesses the purity profile and detects any impurities that might be present. Moisture content is checked using Karl-Fischer titration, or KF, to ensure the sample isn't too damp, as moisture can affect stability and performance. Residual solvents are analyzed by GC, or gas chromatography, to confirm that solvent levels are within acceptable limits, ensuring safety and compliance. We also look at the LOD, or limit of detection, which helps identify trace impurities, and the melting range, which provides insight into the compound's physical characteristics. The appearance of the product, its color, texture, and general look, is also evaluated to ensure consistency and quality. Additionally, if there's any specific monograph test required, we include that to meet regulatory standards. All these tests are part of the qualification process, ensuring the product is safe, effective, and of high quality before it reaches the market. So, in summary, these identification and testing methods form the backbone of our quality assurance system, helping us maintain rigorous standards throughout. Let's now delve into the calculation of assigned potency. This is a crucial step in understanding how we quantify the strength or effectiveness of a substance. Essentially, we start by identifying the relevant parameters, such as the concentration and activity levels. Then, we apply specific formulas to determine the assigned potency, which helps us standardize different batches or formulations. It's important because it ensures consistency, quality control, and accurate dosing. Remember, the key is to connect these calculations back to the overall goal of maintaining efficacy and safety in our products. So, understanding this process allows us to better interpret results and make informed decisions in our work. Let's talk about real-time and accelerated stability testing, especially if it's required for your product. These tests help us understand how a drug substance or product behaves over time under different conditions. Real-time stability gives us data during the actual storage period, while accelerated stability speeds things up to predict long-term stability more quickly. Ongoing monitoring is a crucial part of this process. We need to regularly assess key parameters like assay, which tells us how much active ingredient is present, and purity, to ensure there are no unwanted impurities. Moisture levels are also important because they can affect stability, and physical appearance helps us spot any visual changes like discoloration or crystallization. All these tests should follow the principles set out by ICH-Q1A, ensuring our stability studies are consistent, reliable, and internationally recognized. Ultimately, stability considerations are about making sure our product remains safe, effective, and of high quality throughout its shelf life. This comprehensive approach helps us maintain confidence in our product from manufacturing to the end user. When storing samples or materials, it's crucial to keep them under controlled conditions to preserve their integrity. Typically, this means maintaining a temperature of 2 to 8 degrees Celsius, which is considered cool and dry. Depending on the material, using a desiccator might also be necessary to keep moisture levels in check. It's equally important to protect these items from light, moisture, and contamination, as these factors can compromise their quality. Using amber vials and airtight containers helps shield sensitive substances from environmental exposure. Additionally, maintaining accurate inventory records ensures that we can track samples efficiently and avoid mix-ups or losses. Proper storage and handling are essential parts of our overall quality assurance process, helping us ensure the reliability and safety of our materials throughout their life cycle. Now, let's take a closer look at what each working standard must contain. First, we have the name of material, which is essential for clear identification. Next is the assigned potency, expressed as a percentage, to ensure we understand the strength of the material we're working with. The batch no or WS no helps us track the specific batch or working standard version. It's also important to include the date of preparation, so we know when it was made, along with the expiry or retest date, to confirm its validity over time. Storage conditions are crucial too, since proper storage ensures the integrity of the standard. Prepared by and approved by fields provide accountability and traceability, confirming who prepared and authorized the standard. Lastly, label requirements are vital for clear communication and compliance. These elements together form the backbone of a reliable and consistent working standard, which is key to maintaining quality and safety throughout our processes. Remember, thorough documentation like this supports overall quality assurance and regulatory compliance. When it comes to usage controls, the first key point is to ensure that the issue quantity is only as per the requirement. This means we should never dispense or use more than what's needed for a specific purpose. Next, it's important to record every usage in the WS logbook. Keeping an accurate log helps us maintain accountability and traceability throughout the process. We also want to avoid repeated exposure, which means minimizing the number of times the same item or material is opened or used repeatedly. This helps reduce contamination risks and preserves the integrity of the items. Additionally, we should always aim to minimize opening, just as a way to maintain sterility and control. Finally, maintaining traceability to test reports is essential. This ensures that every usage is linked back to the corresponding test documentation, which supports quality assurance and compliance. Overall, these controls are vital in maintaining the integrity, safety, and accountability of our processes, aligning with our overall goal of quality management. Let's take a closer look at the key documentation involved in the qualification process. First, we have the Certificate of Analysis, or COA, of the working standard, which verifies the quality and consistency of the materials used. Then, there's the batch record, which details the manufacturing process and ensures traceability. We also rely heavily on raw data, such as chromatograms and spectra, to confirm the purity and identity of our batches. The stability protocol and data are crucial for understanding how the product maintains its quality over time. The distribution log helps us track where and when the product has been distributed, supporting traceability and accountability. If there's a need for expiry extension or revision, all relevant details are documented thoroughly. Finally, proper documentation requirements are fundamental to ensure compliance and facilitate audits. These elements collectively support the integrity of our qualification process and ensure we meet regulatory standards. Let's now look at some key principles that ensure data integrity and compliance. First, the ALCOA Plus principles are the foundation for trustworthy data. They emphasize that data must be attributable, legible, contemporaneous, original, and accurate, with additional elements to strengthen integrity. Next, conducting an audit trail review is critical. It means regularly checking the records to ensure all data entries and modifications are properly documented, traceable, and verifiable. This helps us maintain transparency and accountability. Electronic data protection is another vital aspect. We need to safeguard data from unauthorized access, alterations, or breaches. Implementing strong security measures ensures that our electronic records remain secure and reliable. Cross-verification with RS data is also essential. By comparing our data with the raw source data, we can confirm consistency and accuracy, preventing discrepancies that might compromise integrity. Additionally, no unofficial or notebook standards are allowed. All data must be recorded according to established procedures and standards, avoiding informal or handwritten notes that could undermine data reliability. Finally, maintaining data integrity compliance means adhering to all these principles and practices consistently. It ensures our data remains trustworthy, compliant, and fit for regulatory purposes, which ultimately supports the overall integrity of our quality system. Now, let's talk about some common errors we see in working with WS. One major mistake is using WS without proper qualification. This means we're not confirming that the WS is suitable for the intended use, which can lead to serious issues. Another mistake is no linkage to the RS batch, so if there's a problem, we can't trace it back to the original batch easily. Incorrect potency assignment is also a common error, where the strength is not properly determined or recorded, impacting the effectiveness of our processes. Improper storage is another critical point. Storing WS in unsuitable conditions can degrade its quality. Missing expiry or label information is a big concern because without clear labeling, we risk using outdated or incorrect materials. We also see a lack of stability follow-up, meaning we don't monitor how WS holds up over time, which can compromise safety and efficacy. Using expired WS is another frequent mistake. Using materials past their expiry date can be dangerous. Overall, these errors highlight the importance of strict management and adherence to protocols when handling WS to ensure safety, quality, and compliance throughout our processes. Let's take a closer look at the key focus areas from the FDA and WHO. First, traceability to RS is crucial, as it ensures that every step in the supply chain is transparent and can be verified. This helps prevent issues like counterfeit products or contamination. Next, having stability data available is essential for understanding how our products hold up over time and under different conditions, which supports quality assurance. Complete documentation is also vital. It provides a thorough record of all processes and decisions, which is necessary for audits and ongoing compliance. Speaking of audits, a proper review of the audit trail allows us to track any changes or actions taken, ensuring accountability. Adequate training is another cornerstone. Well-trained staff are better equipped to follow procedures correctly and recognize potential issues early on. Controlled issuance refers to managing product distribution carefully to prevent unauthorized access or misuse. Finally, understanding the audit expectations, especially from a regulatory perspective, helps us prepare for inspections and ensures we meet all necessary standards. Overall, these focus areas are interconnected, all aimed at maintaining high quality, safety, and compliance in our operations. A SOP, or Standard Operating Procedure, is essential for ensuring consistency and quality in our processes. It must cover several key areas to be effective. First, there's preparation and qualification, which involves making sure all materials and personnel are ready and properly trained before starting any work. Then, we have the testing procedure, where we clearly define how tests are to be conducted to obtain reliable results. Calculations are next, and they should be detailed to ensure accuracy and repeatability. Storage is also critical, as it involves proper conditions to maintain product integrity. Labeling is important too, because correct labels help prevent mix-ups and ensure traceability. Distribution covers how the product is safely transported to minimize risks. Requalification is about regularly reviewing and verifying that processes remain compliant and effective over time. Lastly, disposal procedures must be outlined to handle waste responsibly and in line with regulations. Overall, these SOP requirements help us maintain high standards, ensure safety, and support compliance across our operations. Let's take a closer look at some of the key steps in our process. First, there's initial qualification, which is all about ensuring that our methods and materials are suitable right from the start. Then, we have periodic requalification, which helps us confirm that everything remains consistent and reliable over time. Stability monitoring is another crucial aspect. It involves continuously checking that our processes stay within acceptable ranges, maintaining product quality throughout its life cycle. When deviations or out-of-specification results, or OOS, happen, we have clear procedures for handling them promptly and effectively to understand and resolve the issues. Change control is also vital. Whether it's methods or materials, any modifications are carefully reviewed and documented to prevent unintended impacts. Archiving expired workstation data ensures we retain important records for future reference and compliance. And finally, lifecycle management ties everything together by overseeing the entire process from start to finish, ensuring ongoing quality and compliance throughout the product's lifespan. These elements collectively support our commitment to quality assurance and continuous improvement, which are at the core of our overall approach. Working standards are fundamental to ensuring the quality, traceability, and consistency of our products. When we talk about working standards, we're referring to the established protocols and procedures that guide our daily operations. These standards help us maintain high quality throughout the manufacturing process. It's also crucial that we comply with pharmacopoeial and regulatory norms, which are the official guidelines set by authorities to ensure safety and efficacy. Proper qualification, thorough documentation, and correct storage are all mandatory components for GMP compliance. These steps not only safeguard the integrity of our products, but also help us demonstrate compliance during audits and inspections. In summary, adhering to working standards is about maintaining excellence in everything we do, aligning with regulatory requirements, and ensuring we deliver safe, effective products consistently. For further support, please contact. Thank you.